So we are talking about epithelial tissue. When you look at every tissue, or let me say every organ in the body, it has got cells. Those cells are going to come together to form tissues. And for each organ that you have is going to be divided into the parenchyma and the stroma. The parenchyma is a part of that organ that performs the actual function. For example, the liver has got hepatocytes, though that's a parenchyma. And then the rest of the other things are just supportive tissue. Okay, so epithelium is a type of tissue. I've got four main types of tissue: epithelial tissue, connective tissue, muscle tissue, nerve tissue. And the principal functions of the epithelium: number one is covering, lining, and protecting surfaces. For example, the epidermis is epithelium, the skin. The second function is absorption. For example, in the intestines, small intestines, the epithelium there is going to help in absorption. Number three is secretion. For example, most of the glands, salivary glands, lacrimal glands, okay, all these glands are going to be producing their product via epithelial tissues. So these are the principal functions of epithelium. Some epithelial tissues have got some muscle attached to them that will help them in contracting so that they can squeeze out what they contain. We call those muscles as the myoepithelium. Okay, myoepithelium. When you look at tissues, I mean epithelial tissue, you can classify the cells depending on the location of the nucleus. If you have got a nucleus that is elongated, you have got tall cells. If the nucleus is flat, you have got flat cells. We call them squamous cells. Tall cells, we call them columnar. When the nucleus is a sphere, we call those as cuboidal or pyramidal. Now, each epithelial tissue has got these properties or characteristics. Number one, epithelial tissues do not have blood vessels. Number two, they do not have nerve supply. Number three, they have got polarity, meaning that they have got the apical end, which is the top, and then the bottom, which you call the basal end. As you can see, this epithelial tissue here, the top that is near the opening, which you call the lumen, is the apical. Down we have got the basal end. On the side we have got the so we call this the basolateral. It's down on the side, basolateral end. When you look at epithelial tissue, they rest on what we call the basement membrane. This guy here, the basement membrane. Each epithelial tissue rests on this basement membrane. The basement membrane, when you look at it using the transmission electron microscope, it is going to have two layers, the basolamina and the fibrous reticular lamina. So you can see the basement membrane has got the basolamina and the reticular lamina. Epithelial tissue on top, and then the basement membrane, and then connective tissue down. The nerves only end on the basement membrane. They don't reach to the epithelial layer. The basal lamina is just where the epithelium rests. The reticular lamina is what connects the basal lamina to the connective tissue. Okay, the basement lamina itself is further resolved into two structures when you even use a more detailed microscope. Remember, I said that the basement membrane has got basal lamina and reticular lamina. The basal lamina is further split into lamina lucida and lamina densa. Lamina lucida is far on top, and then lamina densa is down. Okay, so the basal lamina has got what we call laminins and type 4 collagen. Reticular lamina has got type 3 collagen. That's what makes it up. When you look at these epithelial cells, they're actually going to be connected to each other. one epithelial cell is connected to another epithelial cell. And there are different things that hold these guys together. The first ones, we call them the tight or zonular occludens. The second things that seal these guys, so the tight or zonular occludens, these will form a seal. Like when you have got one epithelial cell and another epithelial cell, you want to seal these guys, hold them together so that you will not have anything passing between the two epithelial cells. So if you see this, there's one epithelial cell this side, there is another epithelial cell this side, and then you have got these occludens junction. Okay, so the tight junctions. After the tight junctions, now again, when you look at the epithelial cell, the tight junctions are the ones that are topmost. When it comes to connecting two epithelial cells, they are the topmost. And then you have got adherent or anchoring junctions. The anchoring junctions like hold one cell to another cell or hold a cell to the basement membrane. So you have got, for example, the desmosomes. The desmosomes will hold one cell to another cell, while the hemidesmosomes will hold one cell to the basement membrane, which is down. And then you have got also the zonular adherents and then the fascia adherents. So these are the ones so after the tight junctions, you are now going to have the anchoring junctions, also known as adherence junctions. You need to know this order because sometimes this picture you can see it. Okay. And then you are going to now, after the anchoring junctions, you now see the desmosomes, which are the ones that you can see here. After the desmosomes, you are now going to see what we call gap junction. So I've got three types of intercellular junction. Inter means between cells. Tight junctions, anchoring junctions, and gap junctions. The gap junctions are the ones here, and they basically allow the communication between one cell and another cell. Okay. And then you can see the hemidesmosomes, they are down here. They are also anchoring junctions, just like the desmosomes. Again, these are the ones you can see: the adherent junction, the desmosomes, tight junctions, and then the gap junctions. So what happens sometimes is that you can have, let's say for example, the skin, right? The epidermis, the epidermis is part of the skin. It's actually epithelium, and it rests on a basement membrane. So sometimes the epidermis can actually disconnect from the basement membrane, and it will end up forming what we call a blister. A blister. Okay. Gap junctions, these guys help for communication between one epithelial cell to another, and they contain what we call a protein connexin. Now, connexin is arranged in hexameric complexes, like six arrangement of six layer. Not really layer, but the way they are arranged in circular pattern, forming what we call a connexon. For example, you can see these guys are six of them, the way you can see them like that. So when they slide over each other, they are going to open this gap junction for things to be able to pass through. And then when they are closed, they close those openings. Epithelium, you've got some specializations on top. For example, you have got microvilli, you have got stereocilia, and for some of them you have got cilia. And this specialization just depends on the function of that epithelium. For example, microvilli is there on those epithelial tissue that are going to be involved in absorption, because microvilli increases the surface area. Stereocilia, this is more is less common type of these processes found on top of epithelial cells, and they are restricted to absorptive epithelial cells, those epithelial cells that are involved in absorption. Stereocilia is actually specifically found in the epididymis and the vas deferens. you need to take note of these two organs where stereocilia is found. So you can see stereocilia is a bit of long, as you can see, and then cilia is a bit of shorter. Cilia, these are long projecting structures. They are larger than microvilli, okay? And cilia are found, so I've got what we call motor cilia, which is found only in epithelium where they are abundant on apical domains of many cuboid or columnar cells. So cilia is actually going to be found on columnar and cuboidal cells. Okay. So this cilia have got some receptor that is going to help in movement. So take note of that. So as you can see, this is cilia and it has got a certain arrangement here of the microtubules. So the arrangement of the microtubules here is known as the 9 plus 2 arrangement. And why do we call it 9 plus 2? Because like you have got these guys arranged. Let me see if there is another picture. Okay, there is no. If you look at, you have got two on the center, two microtubules on the center, and then you have got 9 groups of microtubules around. If you count these, are going to be 9. That's what we call 9 plus 2. So this 9 plus 2 arrangement is important for the movement of the cilia, because if you didn't have these two microtubules on the center, that's known as 9 plus 0, and such cannot allow for movement. Okay. So there's a question here. Label the following diagram. So you need to label. Remember I said that the gap junctions are in order. So what is 1, what is 2, what is 3, what is 4, and what is 5. You can try out this one and send. We see what you're going to find. Now when it comes to the epithelium, you have got the covering epithelium like found like on the skin, in the GIT, and then you have got secretory epithelium, which are the epithelium that is found in glands. Okay. Now epithelium is going to be classified depending on two things. Number one, on the number of layers that epithelium is going to have, and number two, the shape of the cells. So depending on the number of layers, if you only have got one layer, we call that as simple epithelium. If you have got more than one layer on top of each other, we call that as stratified epithelium. When you look at the shape of the cell, if you have got a flat cell, we call that as squamous. If you have got a cell that is equal in length and breadth, we call that as a cuboid epithelium. And then if you have got a cell that is taller than in the width, we call that as a columnar epithelium. For example, this is a squamous epithelium here. Because these are one layers, each one of them is one layer, so we put simple in front. Simple squamous, simple columnar, simple cuboidal. The columnar is longer up. The cuboidal is equal in size, both length and breadth. But if you have got more than one layer, we call that as stratified epithelium. So because these cells are flat and they're on top of each other, stratified squamous. These ones are cuboidal, but then they're on top of each other, that is stratified cuboidal. And then you have got this one. You can see the layer, this is just one cell, but then it seems as though the layers are a lot, because the nucleus has got different levels at which it is found. So this looks as though it is stratified, but it is not stratified. So we call it is false stratified. The word for false is pseudo, so pseudo stratified epithelium. So you actually need to know where these guys are found. For example, simple squamous epithelium. Simple squamous is, you have got flat cells and only one layer. So you are normally finding them where there's going to be diffusion happening, right? Diffusion and filtration. For example, in the blood vessels, the epithelium of the blood vessels, the lymphatic vessels, the hair sac, so that they can be exchange of gases, the lining of the heart, also which we call the mesothelium. When it comes to simple cuboidal, this one is specifically found in those organs to allow for secretion and absorption. For example, basically in the nephron, the kidney. For example, the kidney tubules, like the distal convoluted tubules, the proximal convoluted tubules, the loop of Henle, and then the ducts of small glands. A gland has got what we call the secretory portion and the excretory portion, but you understand that that's the next topic in histology, so please take note of that. Simple columnar, the one where the cells are longer, they are taller than in the width. So for simple columnar, you have got one that has got cilia and the one that doesn't have cilia. The one that has got cilia, we are going to find it in the bronchioles, that is in the respiratory tract, even in the uterus. I mean, in the uterine tubes, the fallopian tubes, to be able to push the egg, because cilia is important in pushing things. In the bronchioles, the cilia will be pushing mucus. In the uterine tubes, the fallopian tube, it will be pushing the egg, and also in the uterus itself. So we call it ciliated simple columnar. And then the non-ciliated simple columnar, it is important for you to state if it's ciliated or not. The non-ciliated is found in the digestive tract, the GIT, and also in the bladder, and is for absorption. Doesn't have any cilia there. The pseudostratified columnar, this is also known as respiratory epithelium because it's actually found in the respiratory system. Pseudo-stratified columnar epithelium always has got cilia. Okay, you can see on top it has got cilia. Might not be always, but it has got cilia. In the upper respiratory tract, you have got ciliated pseudo-stratified columnar there, and also in the trachea. The trachea is part of the respiratory tract. When you go to the epithelium that has got layers, like the stratified epithelium, starting with the stratified epithelium, stratified squamous, a lot of layers which are flat. Now for stratified squamous, they can either have keratin or without keratin, so that's keratinized and non-keratinized. If you have keratin, just know that that epithelium is found in a dry region, but without keratin it is in a soft region, the one that has got like water. For example, you have got a stratified, a lot of layers, squamous, flat layers, keratinized. There is keratin, so it's supposed to be found in dry areas. So stratified squamous keratinized is found in the skin, the epidermis, okay, the outermost layer of the skin. And then for the stratified squamous non-keratinized, is found in places that are soft, that have got water. For example, the mouth, the esophagus, and the vagina. Okay, that's where you find the stratified squamous. You can even see when you look at the epithelium, look at the layer. The cells on top are not dead. You can see nucleus on top. But if you got a slide for stratified squamous keratinized, the cells on top, you can't see any nucleus there because it's dead. Keratin is dead. Okay, but these ones you can see tumor cells on top. And then for stratified cuboid, these guys are found in ducts of salivary glands and also in sweat glands. For stratified columnar, you'll find it like in the conjunctiva, okay, conjunctiva lining of the eyelids. So it's important that you understand where these guys are found. Of course, you learn more by looking at them more and answering questions more. Where there is also another type of epithelium which you call transitional epithelium. This is where like the epithelium is changing, right? It's changing. We also call it the uroepithelium because it's found in the urinary tract. From starting from the kidney to the proximal part of the urethra, the first part of the urethra. So from the kidney, you're going to have the ureters. From the ureter, you have got the blood, and then from the blood, you have got the urethra, but only the first part of the urethra. This contains the urinary urothelium or transitional epithelium. Okay, this is how it looks like. You can see the layers actually change. The types of cells that you find in urothelium, you have got umbrella cells, intermediate cells, basal cells. So in case they ask you to mention some cells that you find here. So sometimes what happens that one type of epithelium can change to another type of epithelium. For example, I said that in the respiratory system you have got stratified, pseudo-stratified ciliated epithelium. So if someone is a chronic smoker, that epithelium can be transformed into stratified squamous, and the change of epithelium is known as metaplasia. All right. So this table is now summarizing all the epitheliums that we've discussed and where they are located. So I've got some questions that you can try out, and I want you to try out these questions and then share the answers. We see if you'll be able to, we see what you are going to be able to answer. Quite a lot of questions here for good practice so that you just make sure that you've understood the topic. So just one thing before I conclude that I should mention about epithelium. Remember we mentioned the the intercellular junctions, the junctions that hold one cell to another. Okay. So when you look at the intercellular junctions, we said we have got the tight junctions, we had the anchoring junctions, we had also the gap junctions. So when you look at these guys, some of them they have got actin, some of them they have got intermediate filaments. Okay, for example, actin is what is holding them at the end, these junctions. So those which contain actin, you have got the adherence junction and the focal adherent. The intermediate filament holds the desmosomes and the hemidesmosomes. Desmosomes hold the cell to the matrix, to the basement membrane. Okay. Desmosome holds one cell to another cell. So these guys contain intermediate filaments. For example, you can see the desmosome there. For the others who contain actin filaments. Just something else again that I should mention here is for the anchoring junctions. These guys will contain cadherins. Cadherins help to hold these anchoring junctions. Okay, please take note of the cadherins, specifically for what? Zonular adherens. For desmosomes, they contain also a cadherin family of what we call desmoglein and desmocholine. These are the ones that will be able to hold them. And these guys are actually going to be inserting themselves on the cytoplasm side of each cell. They'll insert into, what do we mean by cytoplasm side? Look at the way this guy is. Do I have another picture? Okay, this is a desmosome. When you look at the way the desmosome is, coming in from the beginning, like the cytoplasm part, you have got the desmin, which is this guy. But then as you go more inside, because I mean outside, because this is one cell, this is another cell. We have to attach this one cell to the other cell. So what to attach these two cells will be desmocholine and desmoglein. But then what is holding these guys to the inside of the cell? You have got the desmoplakin, the placoglobin. Okay, those are the ones. Desmoplakin and the placoglobin. Hemidesmosomes also contain instead of containing cadherins, they contain integrins. Hemidesmosomes hold the cell to the basement membrane. So I wanted to mention this. Please take note. Okay. So that is where we end